All right, hello. So this is a presentation of a bit of research that I did in paleoentomology with my good friend and colleague, Zhihong Huang. Say hello, Zhihong.
Hello, I'm Zhihong.
So I'm just going to quickly go through my CV and then introduce myself. And Zhihong's going to introduce himself and go through his CV. So my name is Tommy O'Rourke. I am currently a PhD student at Lehigh University working on paleoinformatics, paleontology, biogeology stuff. I have a bachelor's degree in philosophy with a biology minor from Rutgers University and a master's degree in biology from Bloomsburg University. I'll now let Zhihong introduce himself.
My name is Zhihong. I'm currently a PhD student at ES department, Earth and Environmental Science department at Lehigh University as well. I graduated from Whitman College with a bachelor degrees in chemistry and from Cornell University with Master of Engineering in Geological Science.
All right, so first off, we should disclaim that since part of this project was to distribute this report on social media, and social media, contains our personal opinions on stuff, we are not, by appearing on this project together, we're not endorsing any personal opinions that the other may have and share on social media. This is just a collaboration of research. So the research we've done is in the field of paleoentomology. Paleoentomology is the study of insects that are no longer with us. They can be extinct or just their communities, so fossil bugs, that sort of stuff. So without much further ado, we'll share our presentation and we'll begin. Okay, so the name of our talk is a review of paleoentomology datasets in the Neotoma Paleo-ecological database in 2020 with some preliminary analysis, presented by me, Tommy O'Rourke, and Zhihong, as we've mentioned. So I believe we'll start with an introduction to the topic, and
Right.
Zhihong is going to present this part.
Right. So my main role in this project is to do some basic literature review on some of the introduction of paleoentomology, just to show you what is paleoentomology and what's the development and history and some of the limitation and the application of entomology that used to reconstruct past ecosystem. So the first one, of course, is the introduction part. So basically, paleoentomology is a science to use insects as a proxies to reconstruct past climatic variations by measuring or recording them and to try to compare their variations and the distribution of abundance over time from different natural habitats. And with that, we can compare historical insect fossils assemblages with some modern distribution records to interpret some possible climatic and ecological variation in some specific areas. So within the paleoentomology, beetles are actually widely used for reconstruction of quaternary paleoecological communities, as they are one of the most important and diverse order in quaternary fossil records. So in this research, we are mainly going to look at beetles as the one of the major proxies in paleoentomology to trying to assess the data and to mine some data that trying to look at the potential of beetles and especially the dung beetles in future research. So for insects as paleology proxies, they are really useful. One reason is that they are pretty sensitive to climatic variations such as temperature and wide range of habitats with different sets of environmental parameters. And on the right, you can see a pie chart. This is the chart we excerpted from one literature talking about ecology of Northern America beetles. So you can see the dung beetles is made up around like 5% of the overall beetles composition. And of course, we have other beetles as indicated for different kind of ecosystem landscapes. For example, you have plants and fungus and bark and wood, which made up like around 50% of those. So insects are considered to be a valuable paleo-ecology records due to their great abundance and biodiversity. So as I talked before, so it can represent a wide range of ecological niche. And they're able to be preserved in sediments with either anaerobic or desiccant environment, so they can be really well preserved. So this is one of the advantage to use insects macrofossils as paleology proxy. Then I'm going to talk about a little bit about development of paleoentomology. So it started from 17th century. So basically at that time, the curiosity toward insects and some of the macrofossils of insects began to happen. But modern paleoentomology really begins during 1950s in University of in Bremerton, UK, where researchers started to examine two Pleistocene sites and found potential ability of insects record to reconstruct past climate. So here I attached a picture showing this is one fantastic article about the development of modern paleoentomology. This article describes basically the overall development of insect science in 20th century and 21st century, and talking about some major figures and some major conferences and societies that really studies paleoentomology. And I have listed several current database and tools. The first one is Neotoma Paleo-ecology database, which is free and online. This is the one that we are going to use in this project. And the second one is National Center for Environmental Information. So NOAA, it's not only for insects, but also for a wide range of other climatic and paleoecological proxies. And the third one is specifically for bugs. So it's called bug CEP, which is used in one of the paper that I'm going to talk about next. The fourth one is the USDA National Insect Collection Database. Then I'm going to talk about some of the limitations of insect records. So one of the limitations is that the abundance and distribution of insects depends significantly that is influenced and controlled by biotic interactions such as predation and parasitism. So their distribution may not completely depend on the climatic alteration or temperature changes. And some of the climatic influence on insects are more crucial toward ecotones. That is basically the edge of different ecosystems. So the area of study may be limited, and anthropogenic activities may alter or disturb natural habitats for insects when studying modern ecology insects. So that's for modern ecology, kind of not quite a problem for paleoentomology, but maybe some of the disturbance may happen when you study the late Holocene modern ecology of insects. And the ability of insect assemblages to reconstruct temporary and short-term climate variation may provide less illustrative result with lower resolutions. And then I'm going to talk about some of the case study that we're going to explore in this research. So one of the major components of beetles record is dung beetles. So showing up over here on the graph, on the picture on the right. So we have two examples of dung beetle that was found in modern landscape. So they made up around 10% of terrestrial beetle population, and different beetles represent different habitats and landscapes, as I talked before. So using those beetles assemblages as indicators can be used for forest coverage or vegetation composition reconstruction, and it's also useful for people to reconstruct distribution of herbivores and megafauna during the last glacial maximum and early Holocene. So in this research, we are gonna basically use the data science. Data science is a body of technique to draw conclusion from large sets of data. And we're gonna use the the R programming language using statistics and the machine learning, dynamic system modeling, and other computational techniques from a large database. So here, this is the screenshot of the coding that Tommy did for this project that he will explain later in his section.
Yes, I primarily did the coding for this work, while Zhihong primarily did the background research. However, I've given us sort of joint credit on this because even though I wrote the code myself, he still had a share in the project. So data science is a body of techniques with lots of applications across many fields. And it's using all of these computational and artificial intelligence techniques to draw conclusions from big data. And that is why it's interesting and why we can use it in science to draw these big conclusions from data sets of, say, fossils. So Zhihong, you want to take the slide, or do you want me to?
Oh, you can start talking about the data science, I think.
Right.
Yeah.
So, well, I should then say something briefly about the previous slide. So we talk in data science and computer programming about application programming interfaces, or APIs. These are methods that computer programmers can use to directly interact with data in databases or other programs to bring the data in directly without having to go to the website and download something. So we're going to focus here on a database that we are intimately familiar with in the EES department at Lehigh University, and that is the Neotoma database. Now, Neotoma is the scientific name for a pack rat, seen here. And rather unhelpfully, the API is called Neotoma, lowercase. So when you see capital Neotoma in this PowerPoint, we're talking about the database. When you see lowercase, we're talking about the program. So what we've done, what we've done in this research is I've written a script in R using the API package Neotoma to download data and put it into a format that is common in our applications called tidy data format. And we've done this now for every insect fossil in the database in the United States and Canada. And that's a typo there. It's actually for the last 5.7 million years, not 500,000. And then our goal, having this data, saving this data, having it in a nice, easy to work with format, we're going to try to develop some tools that we can use, some programmatic tools to analyze this data. So here's where we downloaded. And so this map comes with the instructions for the API. So I can't 100% verify that this is what this stands for, but the red is supposed to stand for fossils where insect data was found, and then black is sort of all sites queried in that region. So altogether, we pulled 182 records, accounting for 6,481 insect taxa. So In order to develop these tools, we needed a test case, and we've chosen our test case completely at random. Well, not entirely at random. It helped that it was easy to spell. Um, we landed on the genus of weevil, or the subfamily of weevil rather, Apion, and here we call it my species. Here's an example of an Apion weevil. So the first thing we're interested in is just sort of some simple linear regressions. So counts of fossils against time, and these are all in calibrated years before present, and counts against space, and that's degrees north latitude within our search area. And then, of course, we want to see the effect, how time and space interact to affect the count. So we've done a multilinear regression on count against space and time. And then we're interested in sort of finding a proxy for niche analysis, which is what we would do with an existing species with existing climate data. So one way to do that is to say, well, here's the count of this species, and we'll look across all 6,480 additional species and see if we can do a regression on the counts of all of the other taxa. So we've done that, and we've been able to project where we would expect scarab fossils to show up based on the community, the rest of the insect community at the site. So then, it would be really helpful to begin to find some way to group these insects sort of by their niche. And of course, that's going to be really hard across some 6,500 groups. But one way we might be able to start looking at it is to say, is to do a principal components analysis. Now, a principal components analysis is sort of like using multilinear regression to generate artificial variables, and then you can sort of see groupings that might not otherwise be visible using these artificial variables. That is an incredibly brief statement of what PCA is, but that can start to allow us to look for patterns in the data. And then we're interested in sort of graphing, obviously, these distributions in 2 dimensions and 3 dimensions. So again, this is the test we get for my species, and you can just sort of see. So these are all the places we looked and didn't find one, and then here are all the active sites where we find. Here are years where we found 1 weevil, years we found 2, etc. And then the latitude, right? This band is just places we've looked, and then here's the distribution where we find at least 1, where we find at least 2, and we find at least 3. And then that same data now is plotted in 3 dimensions. So this is the we've looked line and didn't find anything, and then this is the here we've found 1. Zhihong, do you have any comments about these last couple of slides?
Yes. So the previous slide so really shows the most of the counts of my species of interest is majorly concentrated on the recent years. So like really at the top of the graph.
That's true. However, we do have to just sort of keep in the back of our minds that so present 2 billion years ago, 4 million years ago, it is clustered here in the present, but in the present relative to 6 million years. Thank you, Zhihong. So here is just an output table of statistical significance for our regressions. Here are our model statements. Obviously, we've left the co-count off because this table would be like 12,000 columns across. But you can see the linearity with this. And what you're finding is marginal significance here in the effect of space. And if we look at the space coefficient, we can see that it's positive, which means that you are slightly more likely to find fossils of my species the further north you go. So this is the results of that co-count modeling, the one where we'd have 12,000 columns if we put it up there. So again, all this is we've looked at the communities of insects per site, and we've said statistically what is the probability, what is the expected count of my species fossils we should find given the community at that site. And you can see here, so this is the observed instances of my species, and this is the projected instances of my species. And so you can see that these are nearly identical, which suggests that we're finding my species everywhere we would expect to find my species. So before we move on, do you have any comments on this?
I think I'm good.
Okay. So we've talked about the Sandom et al. Paper, the work on dung beetles and how important they are. So we're going to now work on dung beetles and see if we can draw some important conclusions from that work. So here is the same. This is the distribution in space. And you can see that with the exception of that one, they're all below 50 degrees. So that would suggest that they're confined to these lower portions of the map. And then again here, you can see there's some doubling up on the more recent time scale. So one thing I've done is I've just increased the resolution of that. So this is since the end of the Younger Dryas cooling period, 11,700 years ago. And you can see that there were some species, and then they seem to have died off. We're not finding these scarab fossils roughly between 6,000 and I'm going to say 2,000 years ago. But that's not unreasonable because we know that during that period of time, there was intermittent cooling periods during that period during the Holocene. So it's not terribly unexpected. So here's the distribution of scarabs in 3D, and you can see these graphs may be a bit difficult to read because of how it looks, but you can see that there's definitely a period beyond which they don't go. Run the regression coefficients here. Again, space is the only significant interactor, and the coefficient is negative, which suggests that you're more likely to find a scarab fossil the further south in latitude you go. So here is the sort of multilinear co-count stuff that we were talking about. And I think this is our most interesting result. You can see here that there's a lot more, there seem to be more scarabs indicated according to our niche analysis than there appear to actually be in the record. And again, part of this may simply be the dissolution of the resolution of the graph. But it looks like we would expect to find some at some higher latitudes than they do occur. So Zhihong, do you have any opinions on this graph? I mean, it's difficult to perceive. It looks like there's some more north, but due to them not being of the same orientation, it becomes a little bit difficult.
Well, you can definitely see, like, for expected number of the macrofossil record, there's kind of like doing in the predicted modeling, there's like should be more more distribution of beetles record on the northern side, northern part of the North America, the overall North America.
Right, right. So we're going to come back and talk about this in a
But
second.
yeah,
But before we do that, I want to talk briefly about our principal components. So we've got two here that account for about 87% of all of our distribution. And what it's finding is, so this is a genus of fly, and this is a genus of diving beetle. And so the implication there is that these flies and diving beetles may in fact be major indicators of environmental conditions and how they affect the count. But again, this preliminary, this research here, this analysis, the PCA, is just a first step along the road in this analysis of trying to find groups and patterns among the distribution of fossils. So here's where we'll get back to our summary of the Scarabidae. So no scarab fossils have been found above 51 degrees north. And so what are the possibilities? So one possibility, probably the obvious one, is that it was too cold for them up that far north. And that's a possibility because we know that cooling periods negatively affect beetle populations. We've talked about that dip in the dung beetle fossils that are consistent with intermittent Holocene cooling periods. But our co-count models show that we should still expect some fossils to be further north than they actually are. So what are the possibilities? So I think there are two main possibilities, and I think they're both true to some extent. One is just a lack of sampling. So ignoring Alaska, because I don't think any records of dung beetles have been found in Alaska. So look just mainly at sort of continental United States and Canada. There's only been 10 sites sampled with any insect data above 50 degrees north. And only one of those contained a dung beetle fossil, and that was that one at 51. So there's definitely the possibility that we might find some more scarab fossils that far north if we sampled that far north. Of course, it's cold and expensive, so that makes sense as to why there wouldn't necessarily be that much sampling. And the other issue, of course, is that we don't really know very much about taphonomy. Is the scientific study of how organisms die and decay and get buried and get preserved. There's not really a lot of insect taphonomy literature, and most of it is quite old. So it's very difficult to say how well these beetles might preserve, especially in cold environments. You think, well, they might preserve better in cold environments. But it's entirely possible that, like, we know, one thing we do know about insect taphonomy is that insects float. And so if you have an insect that's floating on the top of the water, and then that water freezes, well, they might be more likely to be eaten that way, or just not preserved because they had been caught frozen in that top and it might have destroyed them. So both of those are possibilities, and it's definitely something to look into in the future. Do you have any additional thoughts, Zhihong?
yes. I put some of the conclusion part in the next slide when we're talking about some of the next step, basically next step that, future research that we can do in addition to this project. So basically, we probably need more data from other online data set, because for this research, we only used Neotoma,
Right.
and that kind of limits our the pool of our data. So more data set can be necessary if we really want to see how comprehensively some specific beetles record exist in Northern America.
So you would agree then that our hypothesis would be we would expect to find more scarab fossils if we looked at other data sets that might contain northern latitudes.
Yes, that's my expectation. What we also need to do is trying to look some climate or some of other, temperature data during Holocene, mid Holocene and late Holocene, trying to see the Northern America climate change during that time, to see if that has some potential effect on the distribution of dung beetles.
Right. And I have had this thought in my mind for quite well, for about 2 weeks now, that they're given these gaping holes in taphonomy literature. It may be useful, and I may add it as a part of my dissertation work, to start plugging the holes in this taphonomy literature. Modern experiments, careful measurements of how insects decay in water, in different temperatures, in different pH levels, in different oxygen concentrations, and that may shed some more light on this issue. So these are the work that we used in this project for our research, as well as a few others in the write-up for stuff that we didn't talk about here. Okay, so Zhihong, do you want to give your final thoughts on this project?
Yes, I think this project definitely expand my academic horizon, because I'm come from another field, mainly geochemistry. So this process definitely build up some of my knowledge on not only the paleoecology, but also some of the insects and paleoentomology works that have been done in past few decades. Also, I think for our project, I think it's definitely a pretty powerful tool that to predict some of the potential sites that we can, some of the ecologists that can be, trying to explore in the future, that like may have contain some potential merit in discovering new macrofossils for records for insects that can be used to reconstruct past climatic variations.
Thank you, Zhihong. So my final thoughts on this. So if you're unfamiliar, my master's degree work was in the field of modern entomology. I studied the invasive Asian hornet, what the media has dubbed murder hornets. So I'm familiar with modern entomology, which is why I was interested in doing this project. And then the other reason is, most of us have seen Jurassic Park, and we've seen insects preserved in amber during, the Cretaceous or before. And that's probably what most people understand paleoentomology to be, when in fact it is this rich study that often focuses a lot more on modern insect fossils as proxies for recent climate change in the Quaternary. This is what my lab does at Lehigh University, is we look at reconstructing the environments of the Ice Age. So, I think this is a really powerful tool. It's certainly a thing I want to incorporate into my dissertation work. And if I can, include maybe some wet work doing studies of insect taphonomy, modernizing that field, since all of the studies were done, it seems, in the 60s, that may do some good in this research as well. So I think this is a really promising line of research, and I'm glad I did it. It was really enlightening. So I think we'll sign off here. Have a happy Thanksgiving, everybody. We're recording this 2 days before. And take care of yourselves in the rest of 2020.
Yeah, take care.